A patient comes in with the classic rare disease symptom, whole exome sequencing comes back, no pathogenic variant is found. The patient goes home undiagnosed. This happens constantly because current tools does not look at the genes one at a time, but some disease only appear when two specific genes are mutated together. We are Team BioWizard, presented by Khushi Dungel and Prapti Paudel, and in this 32 hours at the Biohackathon 2026, we built a tool to solve exactly that. Tools like CleanVar, Shift, and OpenTarget built around the single gene analysis. There is no simple live platform that takes a disease and tells you which pair of genes are most likely driving each through digenetic inheritance. The only existing resource is the DIDA database, and it is so small that manually curated and static data cannot be integrated with the live population frequency data. So no constraint scoring or any AI interpretation can be seen. We need something that is automated, multi-database, and clinically auctionable, and that is what we are going to create. The objective of the whole uh, biohackathon of our project is to develop an interactive web application, analyze the gene mutation, protein interaction, disease association, and provide a clear visual and statistical insight and make digenic analysis easy and accessible. For this project, we have used Bardet-Biedl syndrome as our main digenic disease. So let me give you some quick biology insights. What is digenic inheritance? Basically, it means one heterozygous variant in a gene A is seen, but you see no disease. But similarly, one heterozygous variant in gene B, and you again see no disease. But both together, you see a disease. Why? Because gene A and gene B are often physically interacting protein in the same biological pathway. Individually, each half loss is compensated. Together, the pathway collapses below the functional threshold. Thus, this is called as the synthetic haplo insufficiency. Our showcase disease is Bardet-Biedl syndrome, as I already told, a ciliopathy where proteins form a complex called the BB-some. When two BBs as genes are each heterozygously hit, BB-some assembly fails, causing retinal degeneration, obesity, and polydactyly. Identifying which pairs matter is critical for diagnostic sequencing. So, what is the main solution that our project gives is that we built the digenic disease architecture explorer in the Python using the Streamlit. We input the disease name. It queries the 4 live databases simultaneously, open targets for gene association, Genomad, V4 for constraint data, string for protein interaction, and reactome for the pathways. It computes a population adjusted digenic score for every interacting gene pair and outputs a ranked AI interpreted report related with the datasets. For the example we have taken, which is the BBS, 9 genes were harvested and 29 interactions were mapped, which is protein-protein interactions, and 29 digenic pairs were scored. Top pair were the BBS9 and BBS4 with a perfect score of 100. Now let me show a demo of it. This is our overview of our app. And now in this overview, we can see that the system is now querying open targets GraphQL in the real time, pulling Genomad constant score, building a string interaction network and running reactome pathway mapping all simultaneously. And here in the overview, we can see the 9 genes, 29 interactions. Now let us go to our different function of the app, which is the gene analysis. Here, we can see that we can see a chart. This chart the chart shows the Genomat PLI score for each BBS gene, which tells us the probability of being intolerant to the loss of function. Red bars above 0.99 mean the human population cannot tolerate even one damaged copy without the consequences. BBS1 at 0.998, BBS110 at 0.99, and CEP90 at 1.00. ARL6 at sign at 0.923, and so on, which is consistent with its role as regulatory modifier, not a core BBSOM structural component. Every single gene passes our PLI greater than zero point 0.9 filter. This basically validates we are working with the right gene. Set for the digenic analysis, the detail card on the right cross validate each gene against the open target association score. Now, let us go through the PPI interaction, which is the protein-protein interaction of the network. Um, from this protein-protein interaction topology, we can see that each node is a BBS gene and each edge is a protein interaction confirmed by the string at confidence over the 700, which is at least 70% probability of being a true biological interaction. If we look at the topology immediately, there is a dense, highly connected core. These are the BB-SOM structural proteins. So even we can even see about our protein interaction by removing or adding any other kind of genes like this. Now let us go through our another property, which is digenic scores. The digenic scores, we see the digenic score. We can see the digenic score of the different types of gene. For example, let's see of ARL6. Um, here it directly shows us the digenic score ranking for different skin. Now let us see the heat map for the same gene. We can look at it, and a whole table for how it works is shown, which helps for the analysis for the researcher and the scientists. Now, here is the interpretation, which is taken by the real data sets, which basically helps doctors to read and interact about the genes, how it affects what happens. It also talks about some of the clinical significances. Also, AI has also interpreted the data, so it might not be fully 100% correct, but most of the data is correct because databases are used. Now let's see, this is another property, which is disease explorer. Basically, we can see the gene analysis or protein interaction of any kind of rare diseases that is found. We can see we can see about any kind of gene from the above given examples. Let us see of the Joubert syndromes. Yes, here we can see about the PPI networks. We can even zoom in and zoom out. There are various kind of features that we have added here. So yeah, this is all about, and this all helps us for the protein-protein interaction, all helps for the so I just zoomed in. These all things helps scientists and doctors to know about these rare diseases. This will help and ease for a lot of things. So that's why we had developed this uh, application. Now, this is the pipeline for our web application, which is first we harvested the gene, then constant filtering was done, PPI network was made, pathway analysis was done, clinical evidence were taken from the different data sets, digenic scoring, as we saw, the heat maps, the bar graphs, everything was done, and AI interpretation, probably with the data sets such as CleanVar and my gene plus were taken. Now this is the digenic score formula that we used. Hope this helps. Um, Now, so that's what we had done. This is the pipeline I just talked about. The analysis and everything was done. This is just a short review. Thank you.